back to your seats. It's very nice to have a coffee break, but it's always hard to get the people back. So now we will hear from the industry side, and the heading here is Developments in Cell Culture-Derived Foods and Food Ingredients. We have 2 speakers also for this session, and the plan is that they should speak around 15 minutes each. And the first one out is Valeria Telloni. She's from Cellular Agriculture Europe, and she is also regulatory affairs professional at Mozambique in the Netherlands. And the title of her talk is Safety Aspects of Cell-Cultured Food, Insights from the Industry. Please, you have the floor.
Thank you. Thank you for the opportunity, and good morning to the audience here and online. It's a pleasure for me today to be here to present on behalf of Cellular Agriculture Europe. We are a trade association based in Belgium, and we are 13 and counting companies producing a variety of cell-based foods, from meat to fish to foie gras and even milk. We are working together to, and I apologize because the layout of the presentation is a bit off. I'm not sure why, but it's fine. Just bear with me. Sorry about that. And we work together in order to have a more resilient and sustainable food system. We have already heard this morning in the previous presentation that sustainability will be a key element for this industry. In order to better advance cellular agriculture at a global scale, we recently entered a collaboration with other cellular agriculture associations around the world. Specifically, last year in October, an alliance was formed between the American Association and the APAC Association in order to achieve a better working together on, we have heard about regulatory harmonization, and where that's not possible or not relevant, we are working on coordination, which is very important, and also to advance sustainability, but also consumer education. Again, this morning we heard from Mr. Sanaha how important will be to educate customers about better and healthier and more sustainable foods. Is the concept of growing meat from or fish or other foods from cell instead of using the whole animal was born in Europe. So in 2013, we presented the proof of concept with this first burger that took a lot of work, a lot of labor, 2 years of work, and it was way more expensive than it is today. When the burger was introduced to the world, we kind of predicted that it will take 10 years before having a consumer product. And we are now seeing more products being approved, and in some cases even eaten around the world. So we hope that this colloquium will really that we can really contribute to have as clear as possible scientific advice that will allow these products to be also in the European markets and not only outside of Europe. As I mentioned earlier, and as it was also mentioned during one of the other presentation, cellular agriculture can really offer a positive contribution to many pillars of the farm to fork policy, EU policy, and also is a boost and can contribute to a resilient supply chain. So we are really hoping to see that we can really contribute in practice more in the future to EU policy goals. Moving now a bit more into the products, as it was already mentioned by Masami in her presentation, there is actually quite a big variety of products and processes, all within the same framework of growing food directly from animal cells in a controlled environment, but there will be differences. So what to expect when the first dossier will end up on your table, that is something to keep in mind, and you have here some impressions of how these foods look like today. So this is a sort of, and this is looking okay, great. So this is a kind of overview of the process. So this is just like a generic depiction of how cell culture works. It kind of mirror already what you have, for example, in the FAO report. So you will be able to make connections. And we start, oops, sorry, I'm wrong. Here, this one, it's very small, I hope you can see it. So we start here from different animals. We select the cells. They can come from a tissue, from a fertilized egg, different types. You get your cell, and then it's the moment to proliferate your cell. In order to do that, you need growth media, which these days are most plant-based media. So, and I come back to that. And you need to refresh the nutrient into the cells in order for them to proliferate. And there was one question in the survey, and something that I hope we can pick up during the break sessions, for example, about recycling media and how there could be any hazards in these steps, for example. Also in growth media, you will see there are a lot of novel ingredients. That's why we are a novel food. So that's also something that we will discuss during the breakout session. Then we move into tissue maturation, and also here you might have a variation between companies. This might be there in different forms, but cells are matured into the cell type you are growing in that specific case. You have your product development, and very important, the analysis of your product. That will also be key to determine the safety of the product. Also, the length of this process can vary quite considerably depending on the cells you are growing. They don't all behave the same, whether it's a mammals or avian or fish. So the key here is, it's the same framework, but expect variation still in the process. Of the potential hazards that were identified, for example, by the FAO reports and other sources, we have decided to have these main subjects for the discussions in the breakup sessions later today. So as I mentioned already, and it was mentioned, various species and cell types. So various species, there might be, for example, different potential diseases depending on the species. So how there will be, for example, list of specific diseases that you want us to test for example, depending on the species or the cell. In regard to selecting your cells and optimizing your cell line, you might have conventional selection methods, like breeding normal animals, like we have been doing for countless years with the conventional species. Or there could be deliberate editing of genes, and of course, like Bruno mentioned, those will follow the relevant regulatory path for evaluating those. Growth media, the good thing about this system is that you know everything that goes in. So we know what goes into the system. So when it comes to toxicity, for example, you can evaluate the toxicity of your raw materials, and if that is not possible, we have some ideas how to do that, and we can, again, discuss this later during the breakout session. Some of the raw materials are novel. Maybe they were never used in food before. There could be like growth factors, for example, that helps the cell growing like they would do in a body. So they need that to grow outside of the body. Cryopreservants, for example. So how do we evaluate these materials? The cell culture process, there could be differences also there. I'm mentioning here scaffolds, for example. Some companies use microcarriers, some others don't. You might have aggregates or just cells grown in suspension, for example. So these will need to be evaluated differently. Then, of course, that's a topic that is came up in the survey, and I expect to be discussed during the breakout session, is genetic stability, how we can ensure that the cells don't go through any change in the culture and processing, and there are some tests that you can do about that. Last but not least, analytics. So this is fat, cultivated fat, 28 days old, I believe. So we need matrix-relevant methods. So I know there are some analytical labs here today, and I'm sure online. So we need methods for analyzing, testing specifically our samples. The media components, not everything that goes into the system necessarily ends in the final product. These are processing aids that have the function to grow the cell and mature the cell, but not everything needs to end up in the final product and might be below the residue levels. And last but not least, also a topic that came up this morning already is the nutrition. So this will also be something that will be addressed, and every company, every product, every cell will have its own like fatty acids composition or amino acids. Are all essential amino acids there, for example, and so on and so forth. So these are not all, but the main elements that we would like to focus on the breakout sessions today. And we are aware that you have different layers of control. So none of these elements alone will ensure the safety of the product, but it's kind of all layers together will give us a safe product. So what the future looks like. So the a fit-for-purpose pathways and dialogue are really key to create a consumer product. As an association, we have experience with pre-submission consultations with other agencies, and those have been really helpful and to learn about the safety of our products. Scaling up and cost reduction is another thing that will for sure happen in the near future, and we are actively working on it. So the cost reduction will see things like removing expensive components, moving to feed-grade material, and which will allow also to scaling up and have a consumer product that is accessible to several people. Finally, there will be still new science and developments coming. For example, when it comes to texture and taste, and the let's say the coordination with consumers association, for example, and other consumer-based elements will be very important to this part. This is also muscle tissue, 7 days old. So how do we move from this to this? And that will also be part of our journey as an industry. This is my last slide. So how can cellular agriculture contribute to the fit for purpose, which is really our goal here? And we can give feedback on the relevant guidelines and also digital tools. We've been participating in the webinar you are organizing on these digital tools, and we are reviewing guidelines. As I mentioned, engage with regulators in other geographies and here consumers about not only the benefits of cellular agriculture but also the safety. And last but not least, we bring a lot of knowledge and expertise about the production on a wide range of cultivated products, so we can really answer a lot of your questions. And there are several of us in this room today, so feel free to reach out to any of them if you have specific questions about specific products. Thank you.
Thank you very much, Valeria. The next speaker is Agnieszka. And the last name, I think I leave it to you. I'm very sorry. I didn't practice this. And the title of your talk is State of the Art in Precision Fermentation and the Potential Application of Models to Support Risk Assessment of Products. And you are representing Europa Bio. So please.
This is not my presentation.
That's not your presentation. That's a problem. I cannot handle. Somebody else needs to fix that. So perhaps while they are fixing it,
Or maybe I can already start, and do the freestyle.
we have time to wait for your presentation. I
Yes,
think.
so AGA, so just to skip the difficult consonants put together. I'm originally from Poland, where we put consonants together to confuse foreigners. I studied biotechnology in Poland, moved to Netherlands already more than 10 years ago, did my PhD in systems biology, and I'm a co-founder of a biotech startup in Groningen, and we're part of the Europe Bio as well, which is why I'm representing them today. Yeah, I can do that too. I was just going to give a brief, overview. So I know there's a lot of experts today in the room. I'm not the expert on being in the lab, because that I left when I left the masters. I moved to the informatics, to the computational side of it. So I know there's Nina Aro is going to speak later today, and she will give you the brilliant overview of what actually happens in the lab. While I'll be focusing more on the how models and new computational technologies can support us in both development, but also probably in supporting risk assessment of products. So, yeah, I can stand here. Yes, I'm short, so I usually try. Like now, I can't see the front row. It's okay. So fermentation, of course, is a very much logical choice for us to move forward. We've been using fermentation for centuries. It's not a new technology, but the modern technologies are really enabling us to create things from scratch, to create meat from scratch without having to involve animals anymore, which is, in my opinion, great. But also to produce a lot of food additives, food products, also medical aspects, also bioproducts in general, packaging and such. We can really move ourselves into a new century where we can decrease our impact on the environment. What is important here is for us, DSP, so downstream processing, so really to ensure that we give the pure product is absolutely essential at this point, so especially for food additives, for the food ingredients and such, we really need to put a lot of efforts here, and that's where, a lot of things go wrong, because this is also very highly driven by the regulatory aspects, how pure it has to be to really make that risk assessment, for us well. Okay. Fermentation, of course, but once it's actually purified, very often you can't even distinguish between is it being purified from a plant or from the fermenter. It's the same protein, it's the same compound, doesn't matter. But a lot of money goes there and risks. And fermentation enables carbon footprint reduction and sustainable production of high yields and pure product. We all know that, right? So what has been happening commercially as well. We had traditional fermentation, of course, historically done. Still, there's a lot of things unknown, so things happen in our grandma's kitchen, and there's not much science on exactly how, but we're doing it anyway because it's good for us. Then we moved on to enhanced fermentation, where we're starting to manipulate certain genes, certain flavors, push our microbes to do something that is out of their comfort zone, to improve the texture, health benefits, and so on, or survival, or yields, so that we can produce more. Here we already started using some certain omics to generate the data, to try to understand better what is happening and how we can push our microbes even further. It's like coaching. And then we moved also, there's a lot of aspects in animal feed. There's more, somehow people are more okay with feeding animals the novel foods rather than ourselves. So there's also that. There's different acceptance levels. So there's a lot of applications in animal feed. Also in ingredient production. Now we've been discussing meat and dairy alternatives. I think it's fascinating. It's not my expertise, so I leave it to others. And the single-cell protein, where we are like specifically growing microbes to produce a lot of proteins to be used as also replacements. So you can do it also with yeast and others. For consumption. Commercialization. So actually there is a very recent, I think, Nature Communications review on what has been happening in the world on different products. So top you see like majority of the focus is on the food ingredients, because there we really purify, we have one compound, it's pure, it's safe, it's easier to bring it to the market. And that's where you see the GMO being used also mostly, because we can purify from the GMO. If there's no traces of DNA, RNA, and no traces of cells, it's safe, right? And then there's some feedstock in yellow, and then the single-cell protein for humans in microalgae or mycoproteins and such. And there's a fermented animal product alternatives that are only just starting. So those are the meat alternatives. And as you see, like going from the top to bottom, there's less and less GMO being used because GMO is still scary. And also when I summarize the graph on the right, half of the products on the market are in the US. Europe has 1, maybe 2 products per country. There's very little. And of course, as we're hearing, there's regulations change. The regulations are not there. There is also different cultures we're talking about, different societies we're talking about, and different approaches. So I was surprised Singapore is not here, because it should be, but somehow the reviewer maybe missed it. But Singapore is now like the one up and coming, and a lot of companies are leaving Europe and moving to Singapore, because it's just, you can't wait 5 to 10 years as a startup until your product is approved. So you will die as a company. That is the state at the moment. That doesn't mean we don't have technology, and we have a lot of many brilliant people in Europe. We have brilliant researchers, but we're not there. So something has to be done on both cultural aspects, on how we teach the society to be more open, how we assess this risk, how we bring the products faster to the market, and how we teach people like me how to take the risks and end products into my strain design. Because we are technology-driven people, I have not thought about if my grandma would eat what I produce. I need to be taught. So there's some teaching on both sides. Plant-based is very good for whole organism. However, it is less effective for single molecules. Sometimes you will need to have a huge field just to get a few milligrams of important compound. So it really depends what are you using it for. Cell-based meat, special functionality, looking very much forward to be able to eat one of those, but currently, apparently, they're really expensive. And fermentation. So fermentation is what I'll be mostly focusing on. And that's where we can produce single molecules. We can also look at the biomass as an addition to feedstock, and really big range of different nutritional proteins, lipids, flavor molecules, vitamins, etc. So for me, as a representative of the industry, I take different aspects and different risks into account. As I said, especially startups, we don't have infinite money to R&D. We need to bring the products to the market fast and de-risk as much as possible. For us, so of course the consumer acceptance, economic viability, RNA content, all of these are risks that we take into account already early on. There's also all the future developments of how we can make ourselves even more green and sustainable using waste products, for example, and really join into the circular economy part. So how we do it, we usually early on try to fail at a feasibility part because it's cheap. So we do a lot of research up front to see, is it even going to make it to the market? Maybe the prices just be too expensive to break it at this point. Is the technology there? Maybe we can't even do it yet. Because it's really, really costly to lose here when you're upscaling. So what we're trying to do, take all the risks that we can think of into our models up front and try to fail fast and move on. This will be discussed more by Nina later. There's many metabolic engineering strategies, the old ways, the new ways, and that we don't know of them yet, but still worked on in the lab, new ways of how you can make your microbe do better. So it comes from like even random mutagenesis, where we have the UV, we have push factors, we have accelerated evolution in the lab, where you can pressure, for example, keep increasing the temperature so that your microbes get better and better at higher temperatures, etc. So you can do the selection, but they take forever and they are expensive and you don't have a guarantee. So it's a bit of a trial and error, and not everybody has a luxury to do that. So currently there's a lot of precision methods like CRISPR-Cas, where we know exactly which gene we want to edit. We do it, and then it has much lower effect on the whole genome, on the whole metabolic structures and networks. So we can really use precision into our editing. So those new technologies are very beneficial for us once we understand the microbes. And also there's many different things we can do later, different metabolic tweaks we can consider, using various substrates, of course, mixtures of waste and mixtures of different compounds to make it cheaper, but also good for our microbes. We can do some cofactor engineering, swapping cofactors. There's also non-canonical cofactors that are being used as a promising new aspect. We can reduce the toxicity level, for example, by making sure that our product is secreted in vesicles instead of just secreted out. That's toxicity for our microbes, for example, and then it can be purified. Because a lot of compounds that are good for us are not good for microbes for a reason, but we make them in microbes, so you can see the problem. So we try to detoxify there as well. Purification, of course, with Cas, we can also tweak specifically, upregulate, downregulate, remove certain parts of the network to really funnel the production into where we want it to go. And we can dynamically control the production as well with different stress factors and that we can apply and use. And in all the stages, we're looking to this. So how many different possibilities are there? If you're looking from the very beginning into, you want to design your experiment, you have multiple model organisms, so you can choose between all the different microbial species. There's lots of them. Now it's just 5. You know what metabolic routes you want to take to get into your product. Then you have to look into media formulation. And then what we're discussing today is the regulatory bottlenecks. And currently that's a question mark every now and then, because we just don't know. And then we sometimes hope that we got it right and our assumptions are correct, and that 2-3 years from now, we're not going to have to redo all the experiments again because actually regulation changed. And we need to prove new things or change our design. And there's, of course, the industrial conditions that we know of. But we can't do 30,000 experiments. I mean, some people can, they have robotics, but not everybody can afford those either. So these days, a lot of people go to computational aided methods where you can actually build digital twins of every single step, and take that into a process, and you literally have a virtual lab in a computer using the data from the past to simulate and choose only the best performing, like top 10% best performance, or 2 best performers, and then you only have to do 96 experiments, which is much more doable in the small lab even, to quickly check whether your product is going to be feasible or not. But to do that, we need a lot of data. So how to select this top 10 performance? We do a lot of course, laboratory solutions for that, but it's really all about the omics. And unfortunately, there is not always a standardization that is necessary to sharing the data between each other. So genomics, yes, we now have standards, sort of. Transcriptomics too, when it goes to metabolomics, that's already a no. Because everybody produces their data differently, they annotate the data differently, they use different formats, everybody has their favorite Excel sheet, and then if you try to do a data analysis on that, you spend 5 years cleaning before you can do anything. I've been there. So it's not an easy topic. So data sharing is extremely important for us as a society. Also in the industry, sometimes we have collaborations where we get data, anonymized data, for example, from past scale-ups. And that's beautiful. So I don't need to know exactly what your product was about, but I want to see where it failed, so that I can learn and not make the same mistakes, right? So anonymization and sharing, a fair sharing of data, where we can see what would be beneficial in developing new standards. So there is, of course, a lot of work done now on fair aspects of data, but it's not enough. So here is really, I'm looking forward to talk about it later in the breakout sessions. What can we do to share this data better? I'm more than happy to share the data. But what we realized, we could have our company with just 10 people doing only modeling, but we needed the lab. So we have an extra entire lab space to generate the data that fits, that it's standardized, of the same quality, of the same annotation, so that we can actually do our models. Because otherwise we just get stuck cleaning the data. So this is extremely important for even understanding the omics. Also, so that we always track the same features, the same aspects, that we don't have the missingness in the data, which is also affecting a lot of the analysis. So it also would be nice from the regulatory aspects, if there are like features that you're really interested in, that are really of key importance, for example, which microbial metabolites are really important to assess the risks and hazards. They should be routinely tracked, and that data should be, available. We shouldn't be guessing what could be potentially harmful or not, especially if there's already knowledge in the field. So there are ways to start, and it's never going to be perfect, but definitely there's a way to go. So because of these large omics data sets, once you have them cleaned up, you can build models. So now we all hear about ChatGPT, great things. It lies a lot, but also sometimes it doesn't. It's important to know when it doesn't or when it does. But we can build these models based on our own data that is specific for fermentation, that is specific for food products, et cetera, when then the likelihood of lying is much lower, right? Because this is actual true data that you feed to the model. A model can show you whether your experiment is going into the wrong trajectory or the right one. It can also show you potential risk factors in your experimental trajectories, where maybe your microbe is undergoing stress, starting releasing certain factors that might be potentially toxic and harmful. Same goes for the microbial community. So we can learn a lot from the data. Like this. There's also other models. It doesn't have to be large language models. It can be simple linear regression that already tells us a lot about the process. So there's two types people discuss usually here: mechanism-driven models and data-driven models. Nowadays, the whole hype is about the data-driven models, but those are only as good as our data is. And if data has a lot of missingness, if the data is not clean, you get wrong answers. That's just that clear. It can't tell you something that is not there. With mechanism-driven models, it's where we use our mechanistic understanding of the principles of biology, physics, chemistry, biology, biochemistry. You put that into the computer, and then you try to simulate what the effect of the network will be. So there you have a bit more understanding, but again, if we don't know a certain pathway exists, we will not have it in our model. The same goes for when a lot of microbes do not have good annotations. It's very difficult to recreate a good models of those microbes if we don't know which genes are there, or we know there is a gene, but we don't know what it does. Or we know that there are certain metabolites, but we don't know how they're connected because the genes are not annotated properly and not added to the function. So there's still a lot of work to be done, both on the industry and on the academia side, to really get to know what it is that we're working with. But for that, there's a lot of tools that you can use, for example, to assess a protein ligand binding, metabolic response to stress, to genomic modifications, secretion profile predictions. We're using that a lot to see whether our microbes are switching in their metabolic state, of whether they're maybe producing something that is going to cause us an issue in the DSP because it's very similar to the product we want to have and you can't purify. And also media and process optimization simulations. Here it's also doing brilliantly because you can really see the effects. You can see where you can make it cheaper or where you can make it cleaner as well, or whether you can use the waste products in combination to lower the costs as well and make it more circular. For data-driven models, all the AI, machine learning, all the hype, you can also use it in different ways for codon optimization or on predicting risk when it's based on the phylogenetic homology similarities. Let's say you isolated a microbe. It's not well annotated. It's one of the new ones. You don't know exactly how it's going to fare compared to the others. You can actually do the genome sequencing, look at the data, see how closely related it is metabolically to others, known organisms, and therefore you know better what risk it might pose, which class it belongs to. So it also gives a lot of information here. We can look at predictions for protein stabilities, folding, etc. These properties are driven by their structures and such. And then it also allows us to know whether we need to secrete a protein externally or do we need to support it with extra glycosylation patterns and such. What would be the effect of glycosylation patterns? Do we get them in the right places? And the generation of proprietary proteins, protein-specific solubility tags or secretion tags to increase the secretion rates to make sure that it's more clean and easy to purify later on so that we can really ensure the purity of the end product. And with that, I'm just going to let you linger with more questions, and I'll be around, and if you have any questions for me, I'm here.
So, thank you very much. We have time for a few questions here for these two talks. So please raise your hands. Don't be shy. And yes, please, over here. Yes. You will get a microphone and a chance to talk.
Hi, yeah, can you hear me? I'm Mary-Liz from Estonia, and I'm from startup, so we share the same struggles. Actually, I have a question to you. So when you were showing the process of failures, so can you elaborate more the first one, which we have to look under this product market fit, or can you say some more details? Because what we are doing currently is a techno-economical analysis, but what else we should consider?
Yeah, that's a very good question. So actually, we're phasing out of techno-economical analysis because at such an early stage, you're looking at 80% error margin. Basically, you're shooting blind. It's not highly predictable. So yes, it can give you justification if your process is ever going to make it because of the cost prices, but that's about it. For the rest, we're looking more on the scientific aspect. So, for example, I was saying, what would the insertion of the pathway do to the host metabolism? So you can do a lot of these. If you know, we have a range of hosts, so we check host suitability to the product. We're looking at what the end product has to be, so which host is more suitable for different DSP, so downstream processing aspects. We computationally integrate the pathways. We check what are the potential issues we're seeing already early on the responses, if one microbe is going to flare stress or not, or have a cofactor imbalance and needs to be targeted extra, or in one host it might just require your insertion of 2 genes, and in the other 5 you go for the one that it's easier, right, and more controllable. Sometimes there's also aspects of food grade safety, so you prioritize microbes that have a GRAS, for example, status, because you know it's going to be easier for later than something that you just digged out from your backyard. So there's many aspects. We also look at the proteins. For example, if it's about the protein product, you can indeed already upfront look if your protein is going to be soluble, if it requires a secretion tag, if it requires a center promoter strengths. So you can already model. So we do a lot of modeling upfront to really understand our problem and see whether it's even technologically feasible to get there, or is it going to require so much technological advancements that we're not going to get there in 10 years. So, yeah.
Wow, yes, thank you. Well, we have time for another question. Yes, over here. And you have to run with the microphone.
On the back, sort of.
You can pass.
Hi, I'm Glauco Bertoldo. I'm an agriculture attaché from the Brazilian mission to the European Union. I wanted to talk for the first speaker. How you think about how this market is going to develop with the idea of us against them? I conduct the first stage of the plant-based regulations in Brazil, and I don't see how an information that you put in your presentation that the 20% of the meat that comes from Brazil to the European Union is related with deforestation, illegal deforestation, because that's information so wrong that I don't understand why you put it there and how it can help the development of this kind of strategy to get a commercial use of this technology. Thank you.
Yeah. So that slide was about how this industry can contribute to the farm to fork in Europe. So that's our vision, how our product can enter into the market and help to have a better supply chain for some of the commodities. So that's where that slide is coming from. But we have here in the room more people that are involved into the policy making. So feel free to reach out to them to discuss this more in detail.
Thank you. And I think we should also try to focus on safety in this colloquium. Thanks a lot. I think we have to move on because time is always running. The next topic out is technical insights into bioprocess design for cultivated meat. And our speaker here is Marianne Ellis, and she's professor of bioprocess and tissue engineering at the University of Bath. So, please, you have the floor.
Thank you. Good morning, everybody. So in the next 15 minutes, I'm going to give you really a whistle-stop tour of bioprocess design. I'm going to focus on a few key aspects. So actually the slides I've got here, I'm hoping they're going to be available to everybody. There's a lot more detail than I can talk to in this space. So I would also say I'm here with my academic hat on. I'm also co-founder, CTO of Cellular Agriculture Limited, developing essentially second-generation scale-up technologies. But this today really is to go into a few details of what we need to think about from the broad piece bioprocess design. Now, before I do that, I'll talk a bit about supply chain and process design. I just want to put a bit of context around how I talk about those, because actually just coming back to the point of thinking about food security and sustainability, that cultured meat, and I'm going through actually a transition myself in terms of what we call it, cultured meat, cultivated meat is what I'll probably say in the next few minutes. Is part of the broader field of cellular agriculture, and actually what, when we're thinking about circular bioeconomy in particular, how we link these technologies. And the earlier we look for those opportunities, the more opportunities, the more chance we have to really nail the environmental opportunities, food safety, social opportunities, and also for the companies, financial viability. So I'm not going to go into detail because this audience knows these. So I'm going to skip that. The links here will come from how, and we've already seen this from previous slides, but how we look at the inputs, how they link to the bioprocess and the outputs, and I'll pick up on a few other aspects of the outputs I think we need to identify, and recognizing the various steps through how muscle cells grow, because ultimately we're trying to produce an alternative protein, and that actually also comes into ensuring that our consumers know what it is that they are consuming, if that isn't the goal. And I was really pleased to hear earlier talking about transition. So just to say again for context, that in the UK we've recently had a nice chunk of cash for the next 7 years to look at cellular agriculture at large, and how we can produce cultured meat and the precision fermentation products with a triple bottom line process, so considering the environment, the social and the economic structure. It is that transition that is important, and hopefully you'll see why I'm mentioning that now as we talk about what we need to consider for bioprocess design. So what we're looking at here is a little bit, I guess, different to what we've recently been hearing about, is how do we link both tissue engineering cellular agriculture and precision fermentation-based cellular agriculture. They've always been quite separate, and kind of understandably, but now we are recognizing there are opportunities to link both for a circular bioeconomy. So when you're looking at mycelium or yeast, algae as well, not on here, how the growth of that and the technologies can link to production of cultivated meat. And I'm just going to touch on this quickly. So you have these slides, but what we've done is an exercise to look at the relative challenges of both the overall value chain and the underpinning technologies. And actually all of these need to come into play to make this a truly viable industry. Okay, so now we're going to get into the detail a bit more. And so we've already seen an example of this. So what I'm going to focus on, if I can go back. What I'm going to actually just focus on is some of the supply chain, but actually I think I'll talk most about the scaffold because we haven't really heard much about that yet. Bioreactor technology is really my background expertise, but downstream processing, waste valorization are linked, as is the water source. I'm going to try and cover those in the next few minutes. Product is not my area of expertise, but it's obviously absolutely key to how we go about producing what we're going to be eating. Nutritional profile and allergens, shelf life are just examples, and I appreciate it's much more in-depth than that, as we've heard earlier. So first of all, the supply chain, and I think it's worth having a think about where we are now and future opportunities. So for those of you in the cultivated meat space, you're well aware, on the whole, that we are buying supplies from research suppliers, at those costs and at those standards. There are some specialist suppliers. So research cell suppliers could be the likes of Promocell and ECACC. But also, we are seeing some companies coming online supplying cells, particularly directly to the industry. And researchers in particular, if you already have license for animal work, can be taking samples from those animals for lab work, and potentially for companies as well. For media, so we know the cost of media is high. There's opportunity, and people are, of course, looking at in-house formulations, buying the components and making their own versions, and essentially sourced from current research supplies. But I think that's going to change, and that will come very much into the safety considerations. We'll probably see an increase in a range of cell suppliers, but also looking at farms and abattoirs directly as cell sourcing, and putting aside the morals and ethics of who owns what. There are safety considerations that will have to be taken into account there. Supermarkets, we're harvesting cells from, one of my amazing PhD students is harvesting cells from purchased meat, and there's a lot of interest around that. It's a very cheap source, but a lot of considerations around that, and actually more likely to have own animals probably through companies or direct links. Media, I think we're going to see more in-house formulations from specialist suppliers, potentially from local farms and also precision fermentation companies. So we can have actually a really big base of supply chain opportunities, but also can make things a lot more complex as we move forwards. So onto the scaffold. So we haven't really heard much about the scaffold yet, so I'm going to pick up on this. So not all cells in our world, in our space, need scaffolds, and there's quite a big drive, I know, to develop non-adherent cells so that you don't need a scaffold. But at the moment, if you're looking to differentiate and mature your cells to get muscle, to get the protein, then as far as I know, that you do need a scaffold, and that might change, but right now you do. So currently, you buy your materials from research suppliers. You might have in-house research consumables. So here is some grass from literally outside our building in Bath that one of our students decellularized, and potentially from other local sources. So Glenn Gaudet's work in the US, for example, taking basically waste plant and utilising that. Again, we're likely to see specialist suppliers, waste valorisation, whether that is from precision fermentation or from the current animal and farm industry and food systems. Again, precision fermentation, food and abattoirs, and supply chains, delis and restaurants. So all of these sourcing considerations, again, I think are really important. Everybody knows how the supply chain actually is at risk, and we have to make sure we have a secure supply chain. That's going to differ where we are globally. How that's done, it needs to be consistent, transparent and safe. So I think this is quite a big piece that we need to be thinking about. So taking the scaffold one step further, very briefly, how you make the scaffold, it's not just about the scaffold itself that you are growing your cells on, it's about what has been used to make those scaffolds. So one of, our long-standing technology in our labs at Bath are hollow fibres. They're made using a solvent, a non-solvent, and we have to think about the residuals left in there. To decellularize plants, you also need to use chemicals to do that. And say Glenn's group have managed to do this with food-safe chemicals components, but that's also important. There are many, many others, silk microcarriers as well. So we need to think about not just the material itself, but the residuals from there. Okay, so last 5 minutes on bioprocess design. So that's our feedstocks. This is the simplified version. Ultimately, your bioprocess, and this is very generic. Companies who already have their pilot plants and production plants will have a variation on this overall theme. So this is very much an academic exercise. You have your feed tanks for your media components, your heat exchanger, more than likely you'll need to be heating, depends on your cell source, but you're likely to be needing to heat your cell media. Your bioreactors, then essentially what comes out of the bioreactors goes in two directions. With your ultimate design, you have your product, a downstream product formulation, where you're removing the product. Also, we need to be looking at separation for waste valorization and recycling to really basically to nail that environmental impact. And here I guess slightly disagree with the last speaker. I think LCA is actually quite important to help us understand this, and particularly with the technologies that are already known and developed and already in use in industry, which we are, I know companies are currently using. I think there's quite a lot we can get from understanding, or at least informing how we do the lab work to help us make those decisions. Okay, so I'm now going to go through in a little bit more detail. So this is from one of my design groups. So for my academic world, our final year chemical engineers have to do a design project. I've been using this example for some time. Scott Allen was part of this group in 2017, was my first PhD student, so I like to use this example. So building a process is bad enough doing it on paper. It's actually a really big job, as you can imagine. Okay, and yes, there's plenty of suppliers. We've heard, this is not, we're not building the first bioprocess ever. There's a lot of kit out there we can already buy. The difference is that when our students are doing this process, you can basically pick drop-downs for what standard of material and what standard of tank that you need for basically anything else, but not for this new industry. And I would love to get to a point where we have those global standards so that it is easy for everybody to know exactly what piping, what material, what basically what fittings we need that are suitable for our industry as we sit between the food grade, but also recognizing the risk of contamination in the bioreactor. Okay, so I'm going to pick up a few points here. So we've got, let's start at the front end. Okay, so we've got the feed tanks here. Most of us will probably be using pre-made media, but powder are almost definitely going to be what we're using in processes. So you can have your own formulation and add what you need when you need it, which means you need to have water being fed in. This is mains water. The supply of that is going to vary depending on where you are in the world. We in the UK we have plenty of rain, and we have usually we have plenty of rain. Sometimes far too much, but sometimes we don't have enough. So we might have to think about backup opportunities. Turkana, Kenya, virtually no rain, but there is an aquifer. So just two examples, I guess two extremes. We've got seawater, but you need to process it. So thinking about that water and what you need to do to it to feed it into your process to make it suitable for cell culture. You've got your mixer, you've got your heat exchanger, you've got your choice of bioreactor coming in here. I'm then going to actually skip to this part again, because I think it's something that's it's talked about, but I think I just want to really highlight it, is the opportunities of what we do for the waste. So we've got our product coming out, but actually we've got a lot of water. At the moment, we've got actually quite a lot of unused media components. And then we've got the waste products that we want to remove that are actually not good for the cells until they're engineered to a way they can stand more. But at the moment, we need to remove it. So an example that this group did was taking off ammonia and lactate with a series of membrane separation steps and recycling actually pure water back to here. So that system has to be basically good enough so that we get a safe feed of water back through. So that kind of circles all the way back here. What we do with this, lactate is actually quite a high value product. Ammonia, or actually just the waste media, can itself be used as basically as a fertilizer or as a feed, so for perhaps going into anaerobic digestion, feeding insects. But if you want to separate it off, there are opportunities there. But if we are going to be recycling back, we need to make sure that it is good enough to go back in. And again, it comes down to essentially the cost, the environmental impact, and actually the technology capabilities as well. So I think they're probably the main parts. I'm pretty much out of time, and I've just finished by saying, above this process design, we absolutely need to take into account infrastructure, life cycle analysis, supply chain logistics, legislation regulation, but also recognising, integrating into current food systems, both for the transition, but also in terms of the technology, and let's be having good, sensible conversations about how we can actually support our food systems for everybody's benefit. And I'll stop there.
Thank you very much. And actually, we have ample time for questions. So this was a lot for me, and I'm learning. That's very good. We need to be ready to assess these products because we know that there will be applications. Really, no questions or comments? Come on.
So the 12-week course.
Don't think about it being live streamed. It's a 12 weeks.
Maybe I'll
Yeah.
go.
Yeah, please. You have to wait for the microphone, though.
Yes, thank you. That was a lot, indeed. But for me, what is important, and also I have a small team for bioprocess design as well, and we're always struggling with the data availability. So something that you now mentioned, the standardization and data that goes along with the tanks, the DSP process, all the designs. Is there any database or any data source that you could recommend for now, before we have all these standards, that would be reliable for use?
I think not right at the moment. So the LCA work that we do, we're kind of generating our own scale work. I know that the papers that have been done have got the data from companies, but actually how they've got that for obvious reasons is protected. I think now, though, with there's a lot more government funding for open access work coming through, and I think collectively across all the countries that have that, then there's opportunity to be providing this better data. But no, at the moment, it's really not widely available. And I completely get the LCAs have really big kind of sensitivity and error on them for that very reason. I think they can be used sensibly and just keep on adding this data in and recognising that what we're reading, the field's moving so quickly that it's going to change actually rapidly. So I'm hoping we're going to get there, but there's not one at the moment. I'll tell you what there is, though, sorry, just to say one thing through GFI, or at least that's how I got it, is actually there is a really nice database for different companies that supply tech. So they are likely to have information about operating specs, which would be useful from that board.
Okay, we have one more question.
Yes, Jette Young, Aarhus University, Denmark. I was wondering, when you are doing these models for the bioreactors, if it's sort of adherent cells, do you have a sort of a frame within what shear you're working, or do you have a sort of a fixed shear and then looking at how the cells are coping with other stresses in the system?
Yeah, so it really depends. So shear stress will depend on the culture system that you've got. With the hollow fibre systems, the cells are actually decoupled from the shear. So we call it pseudovascularization because the fibres act as blood vessels to feed the cells. So they effectively don't experience shear. You can have low flow around the outside, but you're in creeping flow regimes. There's the opportunity to apply it, but effectively it's zero.
Thank you. Wolfgang Geldmann, EFSA. Where do you see the main challenges? Why such foods, and I'm not talking about animal cell culture-derived foods, have not brought yet to the market in Europe? Why they have not even been submitted to EFSA? What are the main technological challenges? I'm not talking about financial challenges or
really good.
regulatory challenges, but why we have not yet large-scale production
it's a really good
ready
question.
to be marketed.
Really good question. And actually, one of the questions that I was going to ask this afternoon is, this is probably more a personal answer actually, is that our understanding is that we actually need to have the exact process and the exact feedstocks known and tested before putting through for regulation. So given the cost and time it takes to build a process to make your product, that actually people are trying to get that right, and that's what's taking the time. So I think everyone, I guess that I know and speak to, you actually have a working process, but not the one that they actually want to use when it comes to commercialization. So I think I think that's why I think we could effectively put something in now, but we know that the design of our systems are going to change within that 18 months, definitely. So I think it's almost the flip is, are we able to is there an opportunity to regulate particular parts of the process and particular feedstocks so that companies will feel, I guess, more confident in putting through for regulation, knowing that there's going to be changes actually really quite quickly. I think that's the...
Thank you. More questions or comments? Yes, over here.
Hello. I'm Jerome from Wageningen University Research. A lot of the LCA analysis or data that I've seen or publications that I've read focus on energy use as being one of the main issues. So I wanted to know if your research group is working on, let's say, bioprocess design towards more efficient or alternative energy uses?
Yeah, so definitely in terms of efficiency, absolutely key to the process design, fundamental, and not just for this field, always has been. So whether that's the design itself or is looking at something called combined heat and power, where you take energy from other parts of that process or another process and use it. So if you've got hot water waste elsewhere, can you use that to heat your cell culture media? And then in terms of actual energy sources, again, it's going to be dependent on where the process is built in the world. But the work that Hannah Tomisto has done, and we looked at, on our system as the base, we looked at wind and solar, and how much difference that makes compared to, good difference it makes compared to like mains electricity. So yeah, for sure, the energy piece is key. And as we see new energies coming online, because of other technology advances, then yeah, we need to be incorporating those in as well.
Of course, this is important for sustainability, but perhaps not for safety in the first place. Okay, I think we then move on. Thanks a lot. Now I think it's time to wrap up this morning session, and I call for Ana Alfonso. She's the head of the Nutrition and Food Innovation Unit at EFSA. So, and thanks a lot to these speakers.
Okay, so I have the happy part is announcing lunch. But before that, I really want to say thank you to all the speakers. The objective of this plenary session was to give an introduction on the two topics, but also to provide a bit of the context, the background, and I think we all heard really fantastic presentations. Me, for myself, I was thinking, wow, I want to do that. That's what I would like to. But no, I'm Ana Afonso. I'm the head of the Nutrition and Food Innovation unit that together with all colleagues have took the task to organize this. So thank you very much to everybody who helped it put it together. I hope you're enjoying, and you will enjoy even more the discussions that we will have in the afternoon. What I will indicate is lunch is on that side, so that's easy. And I would like to ask you to all be here at 1:30. Thank you very much, Mrs. Chair. She's been doing an amazing job standing up to warn people that they've reached their time, and we are on time, so thank you very much for that. So have a good lunch, and we see you here at 1:30.